Thank you. Thank you, everybody.
Okay, let's get started. So we're continuing on in our red cell metabolism lectures. This is lecture 10. We have studied the red cell membrane. We've studied the red cell composition of the lipid bilayer. And so now we're going to study about the content within the cell, all right? And so what's the number one thing in our red cells? Hemoglobin. So we are actually going to build our hemoglobin in the next 2 lectures. All right, if you think about the hemoglobin structure, it's a molecule. It consists of 4 globin chains. And then the heme component is going to be nestled in a hydrophobic crevice within those chains. And the heme is actually a tetrapyrrole ring with ferrous iron in the center. Remember, we want that ferrous iron ferrous state so it can bind to oxygen. And then each heme molecule will carry 1 molecule of O2. So one red cell can carry 4 molecules of oxygen, okay? You guys remember that 65% of our hemoglobin synthesis occurs during the nucleated stages of our red cell maturation. And then 35% of that hemoglobin is going to be in the reticulocyte phase. All right, so then after that reticulocyte phase, the red cell has all of the hemoglobin that it is going to contain throughout its life cycle. All right, so I mentioned the hemoglobin structure. It consists of globin. And when you think about globin, globin is a tetramer of 2 pairs of unlike globin polypeptide chains. So there's actually 4 globular subunits. And we're going to break this down, and this is going to make more sense to you. And if you think about the contents of heme, you have your globin chains, okay? And then you have your heme molecule, which is what is going to contain the iron. All right, and so what your heme is going to consist of is a protoporphyrin ring. And so we're going to also talk about what is, how that protoporphyrin ring forms and how it's broken down. All right, so this is just a little schematic that might make more sense to you. All right, so remember your hemoglobin molecule is composed of 4 polypeptide chains. Usually you have a alpha chain and then what we call a non-alpha chain. So it can be like a beta chain, it can be a gamma chain, all right? And so your alpha chains, indicated here by the pinkish purplish, will bind to a non-alpha chain. In this schematic, these are beta chains, okay? And so these beta chains bind together, and then notice that in between these chains, here's your heme molecule. So here's your heme, and then that little red dot, that's your iron. Okay, so that iron is trapped in that little hydrophobic crevice. It's protected from oxidation inside the globin molecule. And if you think about what protoporphyrin ring is, it's just a combination of hydrogen molecules, carbon molecules, and nitrogen molecules. Okay, that's your protoporphyrin ring. And notice that iron is right in the middle of that protoporphyrin ring. All right, for hemoglobin synthesis, it is very dependent. It's dependent upon the adequate iron delivery and supply. It's also dependent upon the adequate synthesis of protoporphyrins. And then you also have to have adequate globin synthesis as well. And keep in mind that it is actually the hemoglobin molecules that give our red cells the color. And it's actually what causes the cell to weigh the cell's weight as well, all right? It's that hemoglobin. So there's actually 250 million hemoglobin molecules per red cell, all right? That's why it's the red cells are so good at carrying oxygen. All right, so carrying oxygen is the cell's main function. It's also going to bring CO2 back from the tissues. All right, so what all controls this? It must have ATP, right? The cell's energy is needed in order for this to happen. All right, so remember we talked about the glycolytic enzyme pathways where the cell obtains ATP, that also controls, that energy controls this as well. All right, so you have to have your globin chains, you have to have your heme molecule, and you have to have your iron. And if any of those are missing, you will have a defective hemoglobin. All right, and that will lead to some of our anemias as we start talking. So this is the foundation of understanding some of our anemias, okay? So some of our anemias, what happens if you have a defective globin chain, or maybe the absence of some of our globin chains, that will result in thalassemia. What if you have the lack of iron, which is a very common anemia here in the United States? That will result in iron deficiency anemia. And then also, if you have a lack of heme, we're going to talk about if there's any enzyme deficiencies in the formation of the protoporphyrin ring, that will result in what we call a sideroblastic anemia. Some of you guys might have heard of lead poisoning before. It is a type of sideroblastic anemia because it prohibits that formation of the protoporphyrin ring. Therefore, iron cannot be incorporated into the globin molecule. Okay, so all of these are types of anemias. These are, they cause microcytic cells, very small, right? Because you have abnormal hemoglobin synthesis and also decreased hemoglobin content, so hypochromic, right? So these are microcytic hypochromic anemias, all right, because of that defective hemoglobin. All right, so first we're going to talk about all of these steps in depth. But first we're going to talk about iron. All right, so 2/3 of the body's iron is bound to heme. So 2/3 of your hemoglobin or your iron will be inside the cell in that hemoglobin molecule. There's 1 milligram of iron for each milliliter of red blood cells. So what that means is on the daily basis, your body needs 20 to 25 milligrams for erythropoiesis, for hemoglobin to be incorporated into that cell, for iron to be in that cell. All right, so does anybody know where we get our iron from? From your diet, all right? From your diet. And remember, iron is the one element in our body that is recycled. The body does a very good job of recycling that iron, and we're going to talk about how we recycle that iron. All right, so the average adult has a total body iron content of approximately 3,500 milligrams. All right, 2/3 of that is going to be composed inside that hemoglobin. 1/3 is going to be present as storage in tissue. And that is going to be 90% will either be ferritin, which is attached to what we call apoferritin. So once the iron attaches to apoferritin, it's termed ferritin. And we will see that stored in our bone marrow. Versus hemosiderin is another storage form of iron, but it's a little bit more degraded form, and it is not as readily metabolically active as our ferritin is. So you'll see hemosiderin in your tissues, such as your liver, your spleen. You can see it in the bone marrow too. You'll also see this hemosiderin if you have an excess amount of iron, such as hemochromatosis or iron overload, then we can see hemochromatosis. Does anybody want to take a guess at what stain we are able to visualize this excess iron with?
Prussian blue.
Prussian blue is our iron stain. We will be able to see this with the Prussian blue stain. You'll also have some iron. Some of you guys might have heard of myoglobin, which is contained in your muscles. Your muscles will use some of that hemoglobin as well. You'll also have it in the cytochrome enzymes of your cells as well. But the most of it is going to be in the storage form. All right, I included this image, and this is from your textbook, just to kind of let you guys know how we get the iron. So we get iron from our daily diet. It's going to be absorbed in the duodenum of your intestines, and then it's going to actually carry on into these enterocytes, all right, indicated right here. All right, so we're going from the intestines. Here is your iron in your enterocyte. It's actually going to bind. We have a protein carrier. It's called transferrin. It will bind to the iron for transport in our plasma. And it's going to be taken to the developing red cells to be incorporated into the hemoglobin molecules of our red cells. All right, so we all know that our red cells can survive about 120 days. At the end of that red cell's life cycle, it's going to not be able to escape those macrophages in the spleen. So then the macrophages are going to phagocytize those red cells. They're going to break down. So they're going to reuse that iron. They're going to reuse those globin chains. Those amino acids are going to be broken down and put back into the amino acid pool. And then the protoporphyrin ring is going to be broken down in the liver and excreted as urobilinogen, okay, and bilirubin as well. All right, but iron from the macrophages can be taken back to your liver, all right, and it's going to be reincorporated into red cells, all right. And just a little bit, you guys, if you read your textbook, you might hear about this protein. It's called hepcidin. It is produced by the liver, and in the response, let's say you have a high amount of iron, so then the hepatocytes don't need to absorb that iron anymore. So hepcidin, if iron increases, hepcidin will also increase, and it will prohibit the absorption of iron into the cells. The body has everything it needs, it doesn't need any more. On the counter side of that, hepcidin also, if the body's iron supply is decreased, hepcidin will also decrease, and therefore absorption of iron can occur. All right, so hepcidin is kind of your iron absorption control protein. So your daily iron intake, your absorption and losses, they're very minimal. All right, so usually on the daily basis, you shed about 1% of your iron, and that's shed through sweat, through tears, through your hair follicles. All right, so you're only losing about 1% of iron on a daily basis. And again, that's because our body is going to try to reuse as much as it can. And then that 1% that is lost is going to be replaced by our diet. All right, so this just kind of breaks it down onto what's going on. So you have your red cell with about 2,500 milligrams of iron. You're going to have about natural senescence of red cells. You're going to lose about 1% of those cells. You're going to lose about 1% of the iron. You're going to have that daily intake of iron from your diet, and then your plasma transferrin your body will either store it or it will be reincorporated into new cells, okay? So a healthy adult has a blood volume of about 4,500 to 5,000 mils. On a daily basis, you're going to lose about 37 to 42 mils of whole blood, and that's just from red cell senescence. And so actually 2 mils of blood contains 1 milligram of iron. So you're actually losing about 20 milligrams of iron on a daily basis. So you need about 18.5 to 20 milligrams of iron to replace what is lost from red cell senescence on the daily basis. Again, the majority of that iron comes from recycling, and then some of that will be replaced by our daily diet. And it's again about that 1 milligram per day of iron that we lose from sweating and cellular shedding. All right, so let's think about our diet. The average Western diet contains approximately 6 milligrams of iron per 1,000 calorie diet. So the typical adult consumes about 2,500 calories per day. So we're getting about 15 to 20 milligrams of iron per day. All right, so 15 to 20 milligrams of iron per day. However, only about 5 to 10% of that is actually going to be absorbed. All right, so that, remember, we need that 1 milligram because our body is recycling so much. We only need about 1 milligram to replace the necessary iron. So if you think about that and think about what we normally eat on a daily basis, and even though our diet contains a lot of excess iron, our body is not absorbing it, not all of it, it only absorbs what it needs. But given that, iron deficiency anemia is still a prominent problem in North America. All right, it is one of our most common anemias, iron deficiency anemia. It's actually in throughout the world too, North America and the world. If you think about third world countries where they don't have access to a healthy diet, all right, iron deficiency is a huge concern for them as well. You also think about the population of patients too. Think about your moms who are either pregnant, they're carrying that baby, so they have an increased need for iron. Maybe they're breastfeeding, so they have an increased need for iron. And then also your developing children, they have a higher increase of iron as well. So those are some of the populations of patients where we will see iron deficiency. And that's the reason most of our foods have become fortified in iron. If you've ever heard of baby formula, it is fortified in iron. And that is the reason why, is because they need excess iron. They need more than the normal person does. You might also see some of our cereals are fortified with iron now. Flour has been fortified with iron as well. And that's just to help alleviate the presence of iron deficiency anemia. Okay, so you guys remember I mentioned that we want our iron to be in the ferrous state so it can easily bind to oxygen. Most of our food, especially vegetables, the iron is in the ferric state, all right? So especially if you're primarily vegetarian and you're only eating vegetables, then your iron is going to be in the ferric state, so it's not readily absorbed by the intestines, all right? However, if you eat red meat, some of you guys might have been on the slight anemic side and somebody said, oh, go eat a big steak. All right, that's why, is because our red meats have the iron. That blood that's in the red meat contains iron. It is in the ferrous state. It will be the most suitable for helping treat iron deficiency anemia. It is ready to be absorbed, ready to bind to that oxygen. All right, so red meat is more readily absorbed iron than like our green leafy vegetables. That's another reason why some of our flour and cereal has been iron fortified too. So if you're a vegetarian, then you can also get iron from those sources as well. All right, so if any of you guys are battling iron deficiency anemia or any type of anemia, red meat is a really good source of iron. So we have some compounds that help in absorption. Fructose, amino acids, and ascorbic acid, which is vitamin C, they will help further absorb iron as well. Remember I said that we want the iron in the ferrous state? For those vegetarians where they are eating primarily protein from leafy green vegetables, we have stomach acids that will convert ferric acid to ferrous acid. All right, so that also helps. So a lot of those acids are involved in converting ferric to ferrous, all right? And so that will further help the absorption of iron. On the counter side of that, some phosphates in milk will decrease absorption. So that's oftentimes, if you take a lot of antacids, then your iron absorption might be decreased as well. If you drink a lot of milk, your iron absorption might be decreased as well too. So kind of mentioned this earlier, there are some factors that affect the daily iron requirements. Those younger children who are actively growing, they have a higher requirement for iron. Females that are on their menstruation cycle, they're losing more than that 1% of cells on a daily basis, so they need more iron during that time of the month. Those females that are pregnant need more iron, and then lactating or breastfeeding mothers need more iron as well. So it's actually children and women that are more prone to iron deficiency anemia. And so we kind of already mentioned this in my little chart that I showed you guys. Remember that of the iron ingested, only about 5 to 10% of that is going to be absorbed, right? And we're really just replacing the iron that was in the 1% of cells that have gone through senescence on a daily basis, and then also some of that iron that's lost through sweat, tears, all right? Some of that is just through normal cell shedding. And then again, that ferric is the most common form of dietary iron, which will be converted to ferrous. And as we go through some of the process of hemoglobin molecule, you guys will see that the iron molecule transfers from ferrous to ferrous. It goes back and forth quite a bit. All right, so this is just another schematic where iron is in the stomach. It's going to be absorbed in the intestines. It's actually going to bind with apoferritin. When iron binds with apoferritin, it is called transferrin. Actually, when iron binds with apoferritin, it's called ferritin. Transferrin is our carrier protein. It will transport iron through the plasma. So when apoferritin binds to iron, it's called ferritin. It's like kind of like a storage. So think about apoferritin being like a cage-like molecule, and that iron is going to be inside that apoferritin. And when that binding occurs, it's called ferritin. And then transferrin is the carrier protein. It will bind to iron for transport through the plasma. That comes into play when we start studying our iron deficiency anemias, looking at our iron studies. We will look at the amount of transferrin receptors. We will look at the amount of serum iron versus storage iron and things like that. So our iron studies come into play when we start trying to differentiate some of our microcytic hypochromic anemias. Is it thalassemia? Is it iron deficiency anemia? So we need to look at our iron studies to differentiate those. All right, so in addition to the heme and non-heme iron, all right, so ferrous versus ferric, heme iron is also present in myoglobin. And so if you think about myoglobin, myoglobin is in your muscles, so your muscles need iron as well, all right, so that myoglobin will supply your muscles. However, myoglobin is not as good at transporting oxygen as your hemoglobin molecule is, okay? Myoglobin from the meat, again, that's a very good source of ferrous iron. All right, so this little schematic, it just shows how the iron is incorporated into the globin molecule. So here you have your transferrin. Okay, this is your carrier protein for iron. So transferrin is going to bring iron to the cell. All right, that endosome is going to break up the transferrin and release the iron. And then iron is going to be stored inside the cell as ferritin. Okay, remember ferritin is the storage of iron. And then you have your heme being produced in the mitochondria. You have your globin chains. And your iron is going to be incorporated into the heme molecule. This is occurring inside the cell. Right, so this just lets you know how iron is being incorporated into the hemoglobin molecule. So if you notice here, the iron is transported in ferric state, right? It will be incorporated into the hemoglobin molecule in ferrous state. Okay. Again, it's carried by your protein transferrin. Again, 2/3 of all of our iron is going to be incorporated into that hemoglobin molecule. And then you might see some excess iron within that cytoplasm. Again, that's your storage form, that's your ferritin. All right, so if you think about what heme is a porphyrin ring. It has a single ferrous iron attached, all right? So that protoporphyrin ring is going to form a hydrophobic pocket, and that's where the heme is going to be incorporated into. And that's just because we don't want iron to be oxidized, right? Some of you guys might have seen a rusty nail, and that's just because that once that iron is oxidized, it starts breaking down and forms that rust. We don't want our body's iron to do that, right? So it has to be protected from oxidation. All right, so we have our iron. Now the iron is going to be incorporated into the heme molecule, but what is composed of our heme? Heme is composed of the protoporphyrin ring, all right? So this is going to occur inside the mitochondria of the cell. And so you have a chemical reaction that's going to occur. It's going to kick off the formation of that protoporphyrin ring. It's going to start with glycine, and succinyl coenzyme A is going to produce aminolavulanic acid. Sorry, I have a hard time saying that word. It's kind of a tongue twister. Aminolavulanic acid, okay? So then you're gonna have your AA acid, all right, that's what I'm gonna say from now on, is going to go through a series of enzymatic steps. And these are the enzymatic steps involved in the formation of our protoporphyrin ring 9. You guys will need to, you don't have to necessarily memorize this process, but you need to be able to recognize these enzymes involved in the process, because if there is ever a deficiency in one of these enzymes, then guess what? We will have abnormal heme synthesis, and that will, such as lead poisoning, that will lead to excess of these enzymes, and that we call that a porphyra. So if any of you guys have ever heard of a porphyra, and these porphyras can be seen excreted in the urine, and that's oftentimes how we diagnose them in the lab, all right? And so it's very important that you recognize these enzymes. So it's porphobilinogen. It's going to go through a catalytic enzyme process to coproporphyrinogen, and then coproporphyrinogen is going to go through protoporphyrin ring 9, all right? So that's ultimately what our heme is comprised of. I think I have a better schematic. Okay, this is what exactly is going on. All right, so you're inside the mitochondria where that coenzyme A is going to bind to glycine, producing your aminolavulanic acid. And then here are your enzymatic steps, right? So porphobilinogen converts to uroporphyrinogen to coproporphyrinogen, right? And this is in the cytoplasm of the cell. Then it's going to go back to the mitochondria, and this is your protoporphyrin ring 9 formation, all right? And then ferroketolase is an enzyme that's going to add iron into that protoporphyrin ring 9. So then we have heme and we have iron. Okay, that makes sense to everybody? So these are your steps. If for any reason you had lead poisoning, then it would block one of these enzymatic steps right here. You would have an excess of your porphobilinogens, and that would lead to a porphyra. That is the principle behind lead poisoning, is it prohibits the formation of heme. So then what happens in lead poisoning, you have all this excess iron that can't be incorporated into the heme molecule, right? Because heme is not being formed. And so then lead poisoning is a type of sideroblastic anemia, meaning that you have excess iron. Okay, and that's why we are able to see that on our Prussian blue stain. Okay. So I don't want you guys to get lost in all of these different types of enzymes. I really just want you to be able to recognize porphobilinogen, uroporphyrinogen, coproporphyrinogen as the steps involved in the formation of the protoporphyrin ring 9. Okay.
You know where they're happening?
Just remember that your mitochondria is the powerhouse of the cell. That's where it's initiated. I don't think you need to go too specific on that. But as we start talking more in depth about lead poisoning and other porphyras, you kind of need to know the foundation of what's going on. Why do we have excess iron in the cells? And so, like I mentioned, each of those steps yields unstable substances called porphyrinogens. And then the porphyrinogens are further oxidized into their stable substances called porphyrins. So if there's any enzymatic deficiency, then you'll have a built up, you'll have an excess of those porphyrins. And then once you have your protoporphyrin ring 9 right here, remember your protoporphyrin ring 9 is just a combination of carbon, hydrogen molecules, all right? And that it protects that iron molecule from oxidation. Carbon, hydrogen, and nitrogen, that's what it's composed of. And again, so if you have any excess blockage in those enzymes, you'll have excess formation of one of those porphyrins. And those disorders are termed porphyrous. You can see those porphyrins, they will accumulate in the liver and the bone marrow. They can even travel to the brain. It's very toxic if they break the blood-brain barrier. If they break that, it can be very toxic once it gets into the central nervous system. So those conditions may be inherited. So if you have a inherited deficiency of some of those enzymes, that results in a porphyria, all right, or it could be acquired, such as lead poisoning, all right, is a known acquired porphyria. So if you have a deficiency in one of these enzymes and you have prevents the protoporphyrin ring 9 from forming, then what's going to happen is you're going to have all of that iron that's in the cytoplasm of the cell. It can't go anywhere. All right, so normally it's incorporated into that heme molecule, but if heme is not being formed, then you're going to have aggregates of iron within the cell. And that is what we can see with our staining techniques, whether you're using the Röntgen stain or the Prussian blue. All right. And so if you're using Prussian blue, remember we use the language siderocyte. All right. So sideroblast, if it's a nucleated red cell, meaning the nucleus is still within the cell, but you can see the iron aggregates, it's termed sideroblast. If it's just a mature red cell that contains iron, it's siderocyte. And then there are some instances where you might have a nucleated red cell and the ferritin aggregates align very around the nucleus. It kind of forms a circle around the nucleus. That is termed a ringed sideroblast, right? The iron aggregates ring around the nucleus. And then here is what your iron aggregates, your ferritin aggregates would look like using the Prussian blue stain. All right, so all of these dark blue staining areas, that's your iron, okay? So notice this cell right here where my pointer is, it has a nucleus surrounded by iron, so that would be a ringed sideroblast, okay? I was looking for just a plain siderocyte. I don't see one that does not have a nucleus, but here's one right here where you just have iron right outside the nucleus, so that would be a sideroblast, okay? Again, you would see this in any type of enzyme deficiency in the formation of a protoporphyrin ring 9, and you will also see this in lead poisoning. Okay, so we have built our iron and we have our protoporphyrin ring 9, so we actually have our heme of the hemoglobin molecule. All right, so the only thing missing is our globin chains, which we're going to talk about in the next lecture. And hemoglobin is composed of 2 pairs of globin chains and 4 heme groups. All right, so you have total of 4 globin chains. A alpha and a non-alpha will bind together to form 1 pair, and then you have another alpha and non-alpha that has another pair. So each hemoglobin molecule has 2 pairs, right, for a total of 4 chains. for, yes, they will. Yeah. Anytime there's iron deficiency anemia, a deficiency in your globin chain, such as thalassemia, or any enzyme deficiency present in the protoporphyrin ring 9 formation, that will result in small cells as well. Microcytic hypochromasia. Okay, any other questions? So remember heme, what is heme composed of? Ferrous iron and protoporphyrin ring 9. So heme is composed of your protoporphyrin ring 9 with the iron, ferrous iron incorporated in the middle of it. Okay. All right, so that was lecture.
So for the exam, are you going to have like a trick question where it's like Porphyrias ring 8 or something?
I don't think so.
Like